Pleasure to have here Josh Goldberg, which is also, first of all, a new faculty at the Hebrew University. Well, welcome, and a good friend. And so Josh did his BA here in the University of Physics, then he continued his PhD in the ICMC, and postdoc at University of Texas at San Antonio. Later on, he was a research assistant professor at Northwestern University in Chicago, and then he came here and established his lab, and we'll hear all about his work soon. So Josh has worked on a variety of animal models of Parkinson's disease and other movement disorders, and his work has focused on the physiological mechanisms underlying neurodegeneration and the pathophysiological adaptation that occurs both in global brain dynamics and in the autonomous activity of the neurons affected by these disorders. And then we'll hear today about the Huntington's disease. And thank you, Josh.
Can you hear me? Okay. So Avia, thank you very much. It's a pleasure to be back here. I gave a seminar here about 6 years ago. If I knew you were going to read all of that, I would have written something a little bit more humble. So today I'm going to talk about Huntington's disease. So Huntington's disease is an autosomal-dominant progressive neurodegenerative disease. Its prevalence is about 1 in 10,000 in Europe, similar numbers in the United States. It results from a CAG repeat in the Huntington gene, which makes it, it's one of the polyglutamine diseases. And if you have too many of these repeats, above 36, above 40, depends how other people define it differently, then you start developing symptoms of the disease. And the more copies you have, the earlier the onset and the worse the severity of the disease. It's not, the mechanism isn't clear, but perhaps this polyglutamine promotes protein aggregation and then creation of nuclear inclusions. But this is, it's not yet known. So here's a movie of a young man who has Huntington's. You see in the back here, his brother is filming this, and this is just from YouTube. And he explains that this man he needs his brother to help him do simple things like shaving. And here he's walking down the hall, and this is a typical Huntington's disease chorea. Chorea is a sort of dance, involuntary dance movement. So as you see, he's a very young man. Usually Huntington's disease patients develop it later in life, but sometimes, as I said, when there are many repeats, CAG repeats in the gene, then you can get an early onset, even teenagers. So today we know that the Huntington's disease is characterized by massive loss of cells in the striatum, this little purple region of the brain. And even though the symptoms probably of the disease they start appearing before the actual cell loss. So maybe there's first some sort of functional decline that leads to the disease, but eventually there's a massive loss, decimation of cells. Now, if we look at a very, very reduced circuit diagram, very biased circuit diagram of the striatum, so the main population of cells are these spiny neurons. And there are two flavors, D1, D2, based on what dopamine receptor they express, and they also have different anatomical projections. And they make about 90 to 95% of the cell bodies in the striatum. And these are the cells that are decimated in the disease. Now, the striatum also has many interneurons that make up maybe 1 to 5% of the cells, and one of them is this giant spiny neuron, the cholinergic interneuron. And people have shown that in Huntington's disease, as well as other diseases, the cholinergic interneuron is actually pretty resilient, and it doesn't show any decline. It's not decimated in this disease, and other cases like ischemia. So it's a very, very robust neuron. in terms of this disease and others. So this cell isn't affected, as far as we know, as far as we can tell. This has been looked at several times in the Huntington pathology. So just a brief introduction to these cells. Many of you have probably seen them, heard about them. Some of you may have even recorded from them. So the cholinergic interneurons, as you see, have a very large dendritic tree. They have a very intricate axonal arborization that covers a big percentage of the striatum. And what's interesting about these cells is that they are autonomously active. They fire action potentials autonomously. They do not require input in order to fire these action potentials. In fact, they can display various firing patterns. You can record from a cell like this for many minutes, and for a certain period you'll see it doing the single spiking, and then later it'll switch to some kind of bursting. So these are very typical. It has these variety of firing patterns. Is something I worked on with Charlie Wilson during my postdoc. Now, the important thing is that these cells are the only source of acetylcholine in the striatum. And importantly, the striatum is really the place that has normally the highest markers of acetylcholine in the brain. So it has to be that these cells determine this level of acetylcholine in the striatum. And perhaps their ongoing activity maintains basic cholinergic tone. So why is this important? Because there's been this hypothesis that's been around, I think it was first formulated in 1962, so it's 50 years ago, about a balance between two neuromodulators that in the striatum. So the other neuromodulator that's very highly expressed, and also the highest expression of it in the brain is in the striatum, is dopamine. And clinicians had this hypothesis that perhaps in order for us to have a healthy striatum, we need to have some sort of balance between dopamine and acetylcholine in the striatum. And in fact, the hypothesis continues to say that in Parkinson's disease there's a disruption in one direction. So you have a loss of dopamine. That's very well known about Parkinson's disease, but that's met by an increase in markers for acetylcholine. But conversely, there's also a claim that in Huntington's disease you see the opposite. At least certainly in the early, if you look at models of Huntington's disease in animals, you see this certainly early on, that there's a drop in acetylcholine, perhaps an increase in dopamine. So again, since the cholinergic interneurons are the only source of acetylcholine, they must be involved one way or another in this balance or imbalance. So of course, we wanted. Okay, we know that Huntington's disease is characterized by a drop in acetylcholine markers, perhaps acetylcholine levels. The first thing to check is maybe this can be explained simply by looking at the firing rate, the ongoing firing rate. We said these cells are autonomous, so perhaps there's a reduction. If you look at a model of Huntington's disease in an animal, in a mouse, and I'll show you in a second, then perhaps you'll find that there's a drop in the mean firing rate, and that could explain why there's a reduction in acetylcholine, because the cells fire less, and that would perhaps something to study. However, Mike Levine from California, he published a paper several years ago about this R6-2 model of Huntington's disease in a mouse, and they looked at this question. They looked at cholinergic interneurons, and they found that their mean firing rate doesn't change in the transgenic mice carrying the mutation relative to the wild types. We did the same thing. I did the same thing. We used two different. This work was done in the previous year. We began it in Chicago, and we used two models of Huntington's disease. One mouse is called the BAC-HD mouse, and the other is the Q175. 175 stands for how many repeats of the CAG there are in the Huntington gene in this animal. So the 175 is pretty high. And if you look at both of these models of the disease and you compare them to the wild type, the distribution of firing rates is very broad, and you can't really find any significant difference. So there doesn't seem to be a change in the basal firing rate of these neurons in the Huntingtonian mice versus the wild type. So the next thing to consider is what are the inputs? What are the excitatory inputs to these cells? Because perhaps there's a change in the driving. Okay, they have a basal rate, but perhaps there's some input, excitatory input, that can drive these cells, and there's a difference between the Huntingtonian mouse and the wild-type mouse. So cholinergic interneurons receive two glutamatergic inputs from two sources. One is from cortex, and those synapses tend to be more distal and distal dendrites. And they also receive glutamatergic inputs from the thalamus, from the interlaminar nuclei of the thalamus. So, but we know something about the relative strength of these two inputs. So here in a paper we published a few years ago, this just shows you sort of a characterization of how strong the inputs are. So here up here you see a diagram of a para-horizontal brain slice from a mouse. This is just a regular wild-type mouse. And if you slice it in a certain way, you can preserve this area of the striatum. You can preserve projections both from the thalamus into the striatum and from cortex. And in this experiment, what you can do is you can stimulate with an electrode fibers coming from the thalamus and cortex and see the response of a cholinergic interneuron that's being recorded here. And this is what you see over here. Let's start here in the bottom. So you see with cortical cells, they're actually, you don't see very well, but there tends to be a little bit of a speed up when you stimulate cortical inputs, but not something very dramatic. However, when you stimulate thalamic input, you get this huge burst in cholinergic interneurons followed by a pause. Now this is actually known from work in behaving primates. So this, for many, many years, since the early 80s, we know that these cells in the primates that are called, when recorded extracellularly, they're called tonically active neurons. Again, it represents the fact that they fire all the time. We now know that they can do this autonomously. They have the same kind of response to a presentation of a reward. If you present a reward, you get this burst and a pause, and sometimes followed by a burst. This isn't a very good example of a burst, but you often see this, and many, many labs have seen this. And Chagai, every one of Chagai's students who's recorded from these cells will have seen this, for example, including myself. So however, in this study that was done by a Japanese group, they showed that if you inject muscimol into the intralaminar nucleus, over here, what happens? You lose this pause response. So we know from the slice work that we can get it, and we also know from the in vivo work that this pause that you see in vivo requires thalamic input. Okay, so now the plot thickens because there was a recent paper by Anton Reiner from Memphis. He's an anatomist who carefully characterized cortical and thalamic synapses in the striatum at large, and he found a very, very clear finding that there's a loss of about 40% of synapses in the thalamus. Eventually, there's also loss of cortical cells, but at least in the animal models, this is a Q140, so it's a similar model to the Q175, except there are only 140 repeats. They find that there's this loss of thalamic synapses in the striatum. And as I said, eventually, about a year later, you also start seeing a loss in cortex, but it's not as dramatic. So now there might be beginning a formulation of an idea that we have thalamic input that might be driving cholinergic interneurons, and now we know that there's loss of thalamic synapses. Perhaps this could explain the drop in acetylcholine. So more generally, what we wanted to look at, and I won't be answering that question today. That's something we're working on. Oh, sorry, I haven't shown you the questions. I'm looking at the wrong screen. So the first question is, what if this is true? So we see these. We know that there's a change in acetylcholine levels, and we know that there's anatomical evidence of loss of thalamic input. So the question is can we find some kind of physiological measure of this, see some sort of adaptation in these animals in the cortical and thalamic inputs to these cells? And then again, can this explain the hypocholinergic state? Okay, so in order to answer this question, I've actually focused in this talk, I'm going to be talking today about the cortical input. I've explained to you that we know that thalamic input is the stronger, more important, but we were interested in assuming we lose the thalamic input, or there's a massive loss, what happens to cortical input? How does the cortex respond to this? Is there some sort of compensation? And therefore we decided to actually look at cortical input to cholinergic interneurons in these models. Now we were able to use a cross of both of these models with Thy1 channel rhodopsin mouse. This is a mouse that expresses channel rhodopsin 2, which, as you know, if you flash light on a fiber or on a cell that expresses channel rhodopsin, you can get it to fire. So these mice express channel rhodopsin under a promoter of Thy1, and what happens is that you get channel rhodopsin in cortical fibers, but you don't get them in thalamic. More importantly, they're non-thalamic fibers, but mostly cortical. So what you do in these experiments is you take brain slices from these animals that are crossed. On the one hand, they're a model of Huntington's disease. They're crossed and they express channel rhodopsin. Then you can take slices and simply stimulate cortical input by flashing light onto the slices. And that's what I'll be showing you. Most of the data today is from this method. So the first thing we did was we just wanted to see what's the effect on extracellular firing. So again, you take these cells in slices, you patch onto them with an electrode, but you basically record extracellularly. So what you see here is, there's no scale bar. It's probably in the next slide. I should move it up. So you see a few seconds of firing, and over here there's a few flashes, a train of flashes of a blue light, which activates channel rhodopsin. And maybe you see there's an extra spike here. If you look at the raster of this same experiment, 8 trials, you see maybe there's a few more spikes here. However, if you do the same experiment on the mouse that has the Q175 het, what you see is a much larger response. A bigger spiking is something you also see a pause. Now here I've basically just shown you a population. I've taken all the cells and looked at the population PSPH of this experiment, so you can see that there seems to be a larger response that's tied to the stimulation. Now we also were able to quantify that cell by cell and see that this is a significant increase of about 20%. So the response is usually, let's say, about 5 spikes per second in this little window, and it moves up to about 8. So maybe it's a 60% increase in the burstiness.
Seems also that the variability increases.
And yeah, this. You mean here in between?
No, the population.
So these are just the evoked. This is not the this is just the value of this peak. It's not the baseline. Actually, see, the baseline actually stays pretty much the same. If you average all the cells, you get around 2 hertz, which is really sort of the population, which is typical in the slice. Now, we also were able to look at the other model, and this is where the scale bar is. Excuse me, 1 second. So you see the same thing again in these back HD models. You get a strong response, and you maybe even see some sort of pause. So it kind of seems like cortex is trying to develop this kind of burst pause ability to create a burst pause in cholinergic interneurons. So we became interested in what is the mechanism of this strengthening of synaptic input to cholinergic interneurons. So the first thing to look at is the presynaptic effect. We wanted to know whether cortical synapses, cholinergic interneuron, is some sort of strengthening of a presynaptic effect to strengthen the synapse. And the way to measure that is a very old and well-known technique, is looking at paired pulses. So these pulses are 100 milliseconds apart. Usually people take 50, but with optogenetics, it's probably better to take 100. But it doesn't matter; you get the same effect. So you see that if you give one pulse, a single pulse, and then 100 milliseconds later another pulse, and you record synaptic currents in cholinergic interneurons, you see that the second response is smaller, which represents the fact that these synapses are probably have a high probability of release, and therefore the second pulse is smaller. So we wanted to see whether there's a change. People often look. You'll see this in papers in physiology. They'll look for a change in this ratio, the paired pulse ratio, but you see that really the distribution is always less than 1. Cortical synapses onto cholinergic interneurons are depressing, but there's no change. There doesn't seem to be a change, a presynaptic change. So the next thing we want to do, we want to look at the minimal response, the unitary synaptic response. So in order to do that, that's also a relatively easy experiment. Is you tune the intensity of your light. In my case, it's an LED. You tune it, you lower it enough so you get this sort of stochastic response. Either you have no response or you have a unitary response, and it happens stochastically. And then you can look at the distribution of these responses. But importantly, you see that we're interested in the location, the second modal of this distribution, and you see that there really is no change. This is the distribution of the second modal. There doesn't seem to be the change. The unitary response isn't different either. However, so we're still, as I said, we're still looking what could explain this stronger response in cholinergic interneurons. So the next thing we did was we have this LED that we actually flash light through the objective onto the slice, and we just tune it up. This LED was made by Thor Labs, and it has this sort of, if you increase, the dial goes from 1 to 6, and you get this sort of sigmoidal response. And here we began to see a very dramatic effect. So if you look at the animals with HD, both models, back HD and Q175, you see that there's a much stronger, larger EPSCs into cortical response. And then we're running, okay, we don't know, we don't think it's presynaptic, so maybe it's postsynaptic. So one of the obvious candidates is try to block perhaps sodium channels. And it's very easy to do that by including a derivative lidocaine called QX314 in the recording pipette. So that goes in and poisons and blocks all the sodium channels, the voltage-dependent sodium channels. And when you do that, lo and behold, you get a normalization. And if you look at the distribution, it's very clear that you get this very strong response that's normalized by QX314. So there seems to be some sort of boosting of the response by postsynaptic voltage-dependent sodium channels. Now I'm going to do a small digression because of this crowd that's more computational. This helped me have an intuition of why this is so. I mean, every physiologist knows this, but I was tried to wonder why this happens, and actually it tied back to something I had done a few years ago in a totally different context. And for that I have to just briefly flash the linear cable theory at you. And this is an equation of a cable. I'm sure if you haven't seen it, you will see it during your training here. So you'll be familiar. This is a mathematical description of a long cable that is considered a model of a dendrite. And this is, of course, due to Rawl, and Dan Segev's done work in this field. And then what I did is you just say, okay, let's assume that we have this linear cable. We add on a nonlinearity. So we add a single gating variable, S, and it has a reversal. So, and then you assume that it has this S infinity that looks like this. So you have some sort of nonlinearity. The reversal is some value. And you want to see what is the effect on the cable. Now, under the assumption that the inputs are weak, you can linearize. Again, I'm sure this is something that you will have learned how to do in your courses here. You can linearize the equations and ask yourself, okay, what is the linear response of the system? Even though there's a nonlinearity in weak perturbations, what's the linear response? But before I tell you that, let's just look at this. And I want to talk. There are four different cases of this kind of nonlinear conductance, and this appears in a review by Yossi Yarom from the year 2000, Hachin and Yarom. This is actually, I stole the figure from there. So you actually have four, in this simple model, you have four situations. We're going to assume that the resting voltage of the dendrite is somewhere in the dynamic, where there's a nice slope to this curve, but then you can have four situations. You can have a nonlinearity that activates with depolarization, but its reversal is hyperpolarized, like delayed rectifier potassium current. You can have the opposite. You have a conductance that is activated in hyperpolarized range, but it gives a depolarizing current, like the H current, HCN current. And then you have the opposite cases. You have something that's activated by depolarization, but also has a depolarizing current, and one of these is this persistent sodium current. And finally, you have the opposite. You have something that's activated by hyperpolarization, but gives it potassium current. The inward rectifying potassium current can have that effect. And in fact, when you have a very strong inward rectifying potassium, you can actually have like a negative going all in one event. And this actually happened in cholinergic interneurons. They have these sort of a reverse spike, where the voltage dies down. So you have these four, and the parameter that comes out of the analysis, the linear analysis of this cable, is interestingly the product of the difference between the rest and the reversal of the conductance and the slope. So this product of the difference in the slope can either be positive or negative. It's zero if the cable is nonlinear, and it's positive in these two cases, and it's negative in these two cases. And in order to kind of understand what that means, you can look at the transfer function of this cable. These are I chose certain parameters for this paper, and you see that when mu is positive, you have you get this sort of, it's not very impressive, but you can get sort of a resonance response, a peak. And on the contrary, if you look at the other one, mu is negative, then you get this you get a strong amplification of the low frequencies of the response in the transfer function.
What is the peak?
It's very, very here. It's very small. This is it depends what parameters you use. You can get something stronger. John Rinzel then wrote a paper a few years later that generalized this analysis to multiple peaks and things like that. But this basically, I just showed you this to help you appreciate why you expect this persistent sodium to do some sort of boosting, because the cable with the persistent sodium will boost the input. If there's a distal input, it can cause it to be seen larger at the other end of the cable.
Is it a property of the cable, or is it more general a property of the persistent sodium?
It's a property of the persistent sodium.
So it's just in the point membrane?
I think it will. I don't think this depends on the, but it was just it was an opportunity to, is a model that I happened to solve, and it was, I wanted to show it. It was a way for me to present. Okay, so going back, so now I'm reminding you that we saw, in not in a model, but in a real case, we have a boosting of cortical inputs in the cell, and it looks like it's due to sodium currents. And we know the persistent sodium current is a good suspect. So what I went ahead and did is I wanted to measure directly the persistent sodium current in the cholinergic interneurons to see whether you see a change in these Huntington mice. So here's the experiment. You use a voltage clamp experiment, which means that you patch onto a cell and use a feedback amplifier to maintain the voltage at a level that you want, and then you give these long steps, about they're 800 milliseconds here, to various voltages, and you record the currents. And if you look out here, about 800 milliseconds out, that can be considered the persistent sodium. It's what sodium current is present. Of course, this isn't only, in order to show you that this is sodium, you have to subtract the TTX, do the same thing with TTX. So here you see very clearly, without the TTX, the more you depolarize, the more inward current you get. In neurophysiology, an inward current is a depolarizing current. However, when you put TTX, you get more of a linear response. And when you subtract the two, you actually get the TTX-sensitive current. And out here, you get a measure of the persistent sodium. And if you look at the whole graph as a function of voltage, this is what you see. It activates around -60. And however, if you look at the Q175 mice, there's about a 20 to 25% increase in the persistent sodium current in these cells. Measured, again, this is measured whole cell, and you fill the cell with cesium to block potassium currents. So it makes the cell electrotonically compact. So this should be a representation of more, not just the soma, but at least it's in the soma we see this. You see this increase of persistent sodium. Okay. So now the question is, okay, we have an effect in extracellular recording, a boosting, and we have a current that's upregulated, and we know. And the question is, can we go back to this extracellular recording or to the recording of these EPSCs and apply a drug that specifically antagonizes the persistent sodium? And there are a few drugs like that. None of them are really perfect. One of the more recent ones is this thing called ranolazine, and if you do this experiment, again, you're recording synaptic inputs in response to optic genetic stimulation of cortical fibers. So you start, as you break in, you give the first you see the size, and then you wash on this ranolazine onto the slice, bathing, and you see that with time, about 5 minutes into the experiment, the response becomes smaller, and you see this across. This is already a different LED, so, but this LED goes from 0 to 100. You see that at the higher intensities, the response gets smaller with the ranolazine. So blocking persistent sodium, in fact, normalizes the size of the response.
John, I am a little bit confused. You claim that you change the sodium, persistent sodium. The experiments are done in voltage clamp. No activation of any persistent sodium occur because you clamp the membrane.
No, but okay, that's a good question. I mean, okay, that's a question I expect to get from physiologists. So people are used to thinking about bursts of amplification of synaptic events, and they show them in voltage. That's the right way to do it. I mean, the more you give it distal event.
But if you want to explain this change in amplitude of synaptic current by upregulation of sodium channels, and you do the experiment in voltage clamp, there's no sodium channels are not activated.
So that's not true if you go distally. I mean, we can talk about that. I think.
How you can activate voltage-dependent sodium without changing the voltage?
So I think that you have some sort of a depolarizing event that's distal where you aren't clamping.
So you don't clamp the cell. That's what you want.
I'm not clamping the whole cell. I think. I mean, that's so. We've gone on to do, and we haven't finished doing this, to do this experiment in current clamp to see the boosting of synaptic events in current events. It was a bigger effect seen here, and this is the data I can show. Yeah.
But you also have to control for changing the probability of having action potential on the presynaptic fiber, because it blocks the sodium everywhere, and these fibers might actually persistent sodium for, I don't know, synaptic release of production of action potential. So it might be that you also block some of the.
I mean, I think that.
You just have less inputs activated by the light when you block it.
That's okay. That's something to think about. I mean, but you're saying so. I mean, I think that when you activate the channel reduction, maybe I'm not understanding your correct, you're still going to activate the terminals, and there's going to be release of a synaptic event along the dendrite. I don't think you're not. I mean, that's independent of persistent sodium. That's going to have calcium release, and it's going to activate. So, I think. I mean, but I don't. I mean, this, in terms of the there's no reason to assume we don't have any difference in the expression of channel adoption in these models. It's crossed with the same animal.
No, channel adoption is the same. The probability of release might be changed.
But I think you're active. I mean, you can do this. You can do. I mean, I think you can do this experiment with CTX and get a small response. But you can, if you activate terminals, you'll still get a response. Yeah, Chaim.
Similar in principle to NMDA, regenerative process.
Yeah, I mean,
So, did you check for NMDA afterwards?
These experiments are all done with APV. All synaptic input is DAP5 is a blocker of NMDA channels. All these experiments, maybe I should have said that we block APV and we block GABA in these experiments. There's no NMDA, and also
You're talking about an amplification of the EPSC.
right.
So, what do you mean by blocking?
The NMDA. No, we didn't block AMPA receptors. Of course not. But NMDA, there's yeah, NMDA receptors are blocked in this experiment. Okay, so, and then again, if we do the similar experiment in extracellular, you see the cell initially has a higher firing rate. It slows down, I'll say in a minute, but the response that's the response to the train of blue light pulses, it becomes smaller with time. And the reason the baseline becomes small is because actually when we know about cholinergic interneurons that their autonomous activity depends on persistent sodium currents, as Ben Bennett and Charlie Wilson showed many years ago. So the persistent sodium, definitely if you're blocking persistent sodium with this drug, eventually these cells are eventually going to all stop. But during, if you wait on 5 minutes until it happens, you see that also the evoked response is smaller. Okay, so now we have this phenomenon, and now we're starting to talk about things that we've just begun to do, and we're in the process of doing. Is okay, what is the mechanism? Why is this happening in Huntington's disease? And okay, so what triggers the upregulation, and what is the mechanism? It's very similar questions. I'll tell you what we think, and I'll show you a first experiment that we've done about this. So what we know that cholinergic interneurons express group 1 metabotropic glutamate receptors. And these are GQ coupled, so this is their pathway. And it's known that in many cell types that activation of the GQ coupled pathway will lead to downregulation of persistent sodium. It's been shown in cortical neurons similarly. So our thinking is that in the normal state, group activation is group 1 mGluRs sort of holds down-regulates the persistent sodium in these cells. However, when as I mentioned in the beginning, we know now that there's loss of thalamic input. So the idea is perhaps now when you lose thalamic synapses, there's less activation of these group 1 mGluRs on the cholinergic interneurons, which eventually leads to an effective upregulation of persistent sodium. So loss of glutamatergic input from thalamus will lead. That's the trigger for this upregulation of persistent sodium through this well-known mechanism. So just to show you one experiment from this, we've begun doing this. So here's an experiment. Again, we're looking at this activation of the responses. And we take a wild-type animal, and we put this drug CPCCOET, I don't even know how to pronounce it, that blocks group 1 or mGluR1, and then you see that you replicate this Huntingtonian phenotype. These EPSCs become larger, and you see this across the population of cells. Okay, so this is work in progress, which we're going to do. So what I've shown you, what I've shown you today is that persistent sodium is upregulated in cholinergic interneurons in 2 mouse models of Huntington's disease in the Q175 and the BAC-HD. This upregulation apparently seems to boost the cortical inputs, synaptic inputs. This causes cholinergic interneurons to respond more vigorously. We saw this with the extracellular recordings to synaptic activation. And if you antagonize group 1 mGluRs, in wild-type animals, you recapitulate the HD phenomenon. So it seems that there is evidence for involvement. So what's happening here is that this is what we're thinking, and I've hinted this a number of times, is that we're witnessing an adaptation in these animals to the loss of thalamic input. As you lose thalamic input, therefore the cortex sort of takes over and tries to drive cholinergic interneurons more powerfully. Now, things to do in the future is to do the other experiment, which we're also doing, is to activate mGluRs in Huntingtonian mice and see that it normalizes the response. These experiments are underway. And then, of course, another interesting branch of this whole project is to what actually happens to the physiology of the response to thalamic input. We sort of haven't looked at that systematically. So that's the next project. And then, of course, even under normal conditions in a normal animal, to look at this interplay between thalamic and cortical inputs. What's the role of mGluRs even in the normal state? How does thalamic input sort of gate cortical input by through activation of mGluRs? And other things is this question that you asked about persistent sodium. I've talked about it being in the dendrites. It could very well be in the soma. It could be in the axon initial segment. So I've luckily Efrat Katz, who was Mike Gutnick's student, just joined my lab a few months ago, and she's one of the few people in the country who knows how to do sodium imaging. So hopefully we're going to have an opportunity to look at this using sodium imaging. Now, how much more time do I have? 15. Oh, that was very fast. Okay, so then in that case, it'll give me an opportunity to maybe give an overview of other projects that I'm hoping to start in the lab. Oh yeah.
So
Oh, that one.
Do the physiology at the age where you see a strong phenotype in the license?
No, this is done. These experiments are done about 4 months. The phenotype appears, very. I mean, it's very minor. But it appears around 10 to 12 months. But you see, I mean, the evidence also from the Q140 is that you see this loss of thalamic input early on, about 4 months, and then cortex starts losing more. That was Anton Reiner's. So we're looking at that period particularly. So this is sort of pretty early on, and this imbalance of acetylcholine and dopamine is actually more pronounced in early. Apparently, the opinion is more pronounced in earlier stages. But the drop in cholinergic markers is supposed to happen in early Huntington's disease, or in the models it happens early on. So in that sense, it's the right time for him to look at.
And then the second one is about the expression of the Huntington protein. I thought it was expressed in MSH. Like the primary cells that express mutant protein are the MSH?
That's a good question. I mean, I think it's also cortical. But eventually these cells are lost. So even if one of the kinds of Anton Reiner and Paul Bollum from Oxford is working on this is that Huntington also has effect on thalamic cells, and they're affected. And we're talking about something that probably has to do with loss of thalamic input to cholinergic interneurons that are resilient to the insult. So I don't know if it really matters where Huntington is in the.
To put it in the context of the model.
Okay.
I think it's how this protein will result in.
So I think it's more. I mean, as I said, an effect. This is a physiological response to something that's happening in thalamus, to thalamic inputs, loss of thalamic cells. Well, the loss of synapses, and as I said, that's Anton Reiner's the first paper. Other people are looking at this as well. Chaim.
So I misunderstand the moral message that the upregulation of the cortical input to compensate for loss of thalamic input, is it good for the disease?
I mean, we don't know. I mean, I think one of the things that I've been asked before is just, okay, you see this minor increase in firing in a very small window around an evoked response. How can this have anything to do with the acetylcholine level? However, we have the expert on acetylcholine esterase here in the room. These we know from Chagai's work and other work that these responses in the striatum, cholinergic interneurons responses, are synchronized. It seems like there's a network of cholinergic interneurons that respond to input. If you record from any one of them and you stimulate, in an animal, you show a reward, or all of them respond. So this means that there's going to be a drop in the if there's a pause, there'll be a drop in acetylcholine that's synchronized, and because acetylcholine esterase is very fast, you might these periods, these synchronized periods of drops could have some effect. I don't know if it's if it's a compensation and we have a mechanistic explanation, but I mean I don't know whether it helps or not.
So about 30 years ago, UNIC published a paper in Nature about striatal cholinesterases being dispersed like a checkboard, with some cells reaching acetylcholinesterase, others in butyryl cholinesterase, which is like 20-fold slower in hydrolyzing acetylcholine. So if there is an association, as we predict, in the primate striatum, you should see
different regions.
It
could be. I mean, this is something. It'd be something hard to test. But cholinergic interneurons, I'm trying to remember now from those papers whether they respect those Gore borders or not. I think they do not. I mean, but they also their axons. As I said, their axons, bifurcate immensely. I mean, they release it in all volumes. They release it. So, yeah, I don't know. So if one of my objectives in giving this talk, aside from the excitement, is that I'd like, I'm a new member of faculty here, and of course I'd like to attract students. So I just wanted to if anybody interested, I'd like to just quickly talk about other things we're going to be doing, and these are things that if anybody of you want to come and join the lab, you will be doing So I mentioned earlier on that we talked about this acetylcholine dopamine balance. And as I said, it's a 50-year-old notion. It's sort of vague. And people have shown biochemically that there are these responses, but nobody's actually come forward and said, show me the network, show me the circuit that creates this balance or is susceptible to the imbalance in the disease. And Stephanie Craig from Oxford a couple of years ago published a very, very interesting paper where she showed that if you stimulate cholinergic interneurons and record electrochemically levels of dopamine in the striatum, you see that when you stimulate cholinergic interneurons, there's an increase in dopamine. And then she also showed that if you stimulate thalamic inputs optogenetically, you get this burst, you get this influx of dopamine. Now, how could cholinergic interneurons cause dopamine release? So it turns out, according to this model, they basically hijack the axons that come from substantia nigra. So these are dopamine fibers. They express nicotinic acetylcholine receptors, and apparently when you either stimulate thalamus or directly cholinergic interneurons, there's release of acetylcholine, which then causes the release of dopamine. So this enables you to start sketching out what circuit is responsible for some sort of covariation of acetylcholine and dopamine. So I have a certain hypothesis about how this circuit is constructed. That's one project I hope to be looking at. Similarly, we're just going to be looking. We talked about thalamic input to cholinergic interneurons, and look at the synaptic properties. These are synapses that haven't been studied very much, and interestingly, we've already found some intriguing results. Is that here? This mouse I've shown you. You use these Thy1 mice that you flash late, and here we did a complementary experiment. I injected channel rhodopsin into the intralaminar nuclei, and you wait 2 weeks. The virus expresses itself. You get thalamic fibers expressing channel rhodopsin. And then you can do the same experiment, and you see that you can do the same thing. What you see, first of all, if you look at the black response, which is the normal response, you see, as we said, cortical input is depressing. This is already in current clamp. Actually, thalamic input is facilitating. However, if you simply keep on stimulating, you start seeing that these responses become smaller. So simple stimulation of thalamic input onto cholinergic interneurons causes a weakening of the synaptic responses. And this, I believe, I already know part of the mechanism, which I won't talk about now, but this might be related to the plasticity, the property of these pauses, and the strength of these responses of TANs to stimuli. We know, again, from papers from many years ago, that if you associate reward with a conditioned stimulus, then you can change the property of these pauses. So here's a mechanism. Simply by stimulation of thalamic input, you already see changes in the synaptic response of cholinergic interneurons. So this is, again, something I think very cool. We already have a punch and a very interesting result, and again, something we'll be looking at. And finally, the project I'm focusing now is something I'm doing in collaboration with Mona, who's in the crowd here, and Onit Sharon from Inkarim, where we're looking at, this is a follow-up of a study I published in last year, a year and a half ago, about neurons in the dorsal motor nucleus of the vagus, which are one of the first neurons known to show pathologies in Parkinson's disease. This has been known for 100 years, so we studied them, and we found a direct linkage between calcium currents and a basal oxidative stress in these cells. However, I became interested in the question, after talking about this with Mona and Ronit, about what about another model important model of Parkinson's disease. We now know that these pathologies that appear in Parkinson's disease, called Lewy pathologies, are these intracellular inclusions that are comprised mostly of a protein called alpha-synuclein. So alpha-synuclein has been looking at very closely its relationship to Parkinson's disease, and I became interested in the possibility that perhaps alpha-synuclein causes oxidative stress in these cells, by virtue of the same mechanism, somehow engaging calcium channels and disrupting their activity and somehow leading to oxidative stress. So we're going to use the same tools we had to link calcium channels with oxidative stress and look at specifically at a model of mice that are overexpress alpha-synuclein. So this is again a project that's underway now. Okay, so of course I want to thank that most of the work I showed you today was done with Jim Surma and Mariangela Scarduccio. You saw a picture of her earlier on. She sent me that picture. So we work on this together. As I was leaving Chicago, she picked up these experiments. The work was funded by the CHDI, and currently I'm funded by the Marie Curie grant that just received, and the work on alpha-synuclein is funded by the Michael J. Fox Foundation. So thank you.
Any more questions? So I'll ask you one thing that when you move from the animal models to humans, are there any suggestions how to check these things?
So a lot of these models is first of all move into the other direction. What people try to do is also true with the alpha-synuclein models, to actually take the mutant protein from humans and somehow express it in the animals. And that's one of the models we looked at is like that. However, the problem with these models is that these animals live for a couple of years, 2 years, and we usually look at them. We do experiments in the first half year of their life, and then we kill them. That lifespan might not be long enough to have everything you see in humans. So these animals you don't see chorea or anything, but you do see these subtle motor defects, and you start seeing these physiological effects. I've spoken about the cholinergic interneuron, which is sort of a new vista on Huntington's disease, but there's plenty of work done in this lab and other labs about spiny neurons and their decimation and how synaptic input to them and growth factors. So you just study it. These are the models we are able to look at. It's a smaller time scale, and we try to learn about the disease. But again, that's an inherent problem also in Parkinson's models, is that the animals have a very short life, and some of these mechanisms might require a longer time to cause more serious pathologies.
But it seems that in Huntington there is an optimal disease, because if you expand the triplet to 1,000, so in a human patient, the disease will express itself at the age of 2 years or 3 years, and just to take a mind, it's much longer.
Yeah, I mean, you see people make these variants, but I think, at some point, this is not something I know too much about, but it could generate other problems. Maybe the animals can't be viable, but people have moved, 140, 175. There's plenty of copies relative to humans. I don't think anybody's trying to reach 1,000 copies. By the way, are you saying that as a fact that there are humans who have thousands of copies and they get them at the age of 2?
700, 800, I can remember.
Oh, okay, I didn't know that. I didn't know that. Okay, so it's really juvenile Huntington.
Thank you, Josh.
Thank you.